Okay, guys, welcome to our next lecture. Today, we're going to start talking about protein structure and in a more sort of larger scale way. We've been spending time talking about amino acids and some of their properties. We spent some time talking about the amide bond, how it's formed. And today, we're going to extend that by talking about the next layer, next levels of protein structure. So I mentioned last time that you have a polypeptide or a protein, a really big polypeptide, that the sequence of amino acids um, is, of course, equivalent to that protein's primary structure um, with additional wrinkles at the level of disulfide bonds or other types of post-translational modifications like hydroxylations, things like that, right? All of those things that sort of exist at the level of the amino acids or the amino acid identity and their sequence is wrapped up in the primary structure of a protein. We're now going to extend that, right? So you can kind of think of primary structure as sort of this sort of linear property of a polypeptide. We're going to now extend that up into three-dimensional space, even though amino acids, of course, are three-dimensional, but to the next higher levels of structural organization of a protein. They depend upon that amino acid sequence, as you'll absolutely, as we'll see, but we're going to start talking about some of the properties of these next layers up of protein structure, specifically secondary, tertiary, and quaternary structure, right? Okay, each of these build on the next, right? So primary structure determines both secondary structure and tertiary structure. Tertiary structure determines, in many cases, how secondary structure determines how tertiary structure is built. And all of those then cascade for oligomeric proteins into their construction into quaternary structure. Okay, so as I mentioned before, I'm going to emphasize here again, a protein's function is directly related to this three-dimensional structure, right? Is that form is function, function is form again, right? These things are inextricable, right? So we really need to understand something about how this works. But before we start digging into some of these details, consider an example, right? Specifically, let's think about how the connection exists between protein structure and human disease. Now, of course, we all know that if you have a mutation that makes a protein unstable, doesn't get produced, you don't produce enough of it, it doesn't have the right activity, or it has, a changed activity. All of those things can be bad and can be deleterious and are connected to a whole family, dozens upon dozens, hundreds of different diseases, right? And in the most extreme case, a mutation that eliminates a protein, you don't even necessarily exist, right? It's an embryonic lethal mutation, right? So that's certainly one family of human diseases where aberrant protein structure is a serious problem. But consider this example, one that was sort of recognized by one particular scientist by the name of Stanley Prusiner, starting in the late 1960s, early 1970s, and his work on this ultimately won him the Nobel Prize. Though you gotta know that it was a fight for him because the idea he came up with was quite controversial, right? So here's sort of a summary, right? There's a protein known as PRP. It's also known as the prion protein because of what this protein actually does. It's a protein found in the outer membrane, the plasma membrane of certain types of neurons. What it does, as far as I know, people still don't understand. It's not essential. You can knock it out. It may be important for neural development, but for whatever reason, this protein has a particular characteristic that is kind of scary. And this was identified by Stanley looking at some well-known historical diseases, first in sheep and then in some other organisms. And it was related to the behavior of this particular protein. So in a model set of experiments that really helps Prusner and others nail this down, if you take that prion protein, PRP, and you engineer some mice, which we've gotten very good at over the last few decades, to express this protein to high levels, sort of unnaturally high levels. It doesn't kill the mice, they develop normally, right? Nothing seems to be wrong with them, but at a non-trivial rate, these mice will develop a quite horrifying, debilitating disorder in which the tissue of their brain, cortex and other parts of the brain, literally begin to dissolve. The tissue develops these big holes, these sort of little white spots you see here. Those aren't supposed to be there, right? And the tissue starts to look like a sponge, which is how these diseases got their name, so-called spongiform encephalopathies. And spongiform encephalopathies, in this case, what this is modeling is a human disease known as new variant CJD or new variant Creutzfeldt-Jakob disease, right? And these mice die. It's terrible. They develop severe ataxia, they fall over, they can't move, they shake, and they eventually stop breathing. It's awful. And more or less a similar thing happens to humans who get CJD. Okay, well, you can take the brains of these little mice and you can grind them up, do an extraction and a clarification. Here's the really scary part. If you intracranially inject that into another mouse, they will very, very quickly and much more rapidly than the original mouse get exactly the same disease. And then you can do it again. And the same thing will happen again. It'll keep happening. The incubation time to getting the disease is directly proportional to the amount of this material that you then inject. It's the direct relationship. And here is the really scary and surprising part. If you take this brain tissue you've homogenized and you treat it with things that destroy nucleic acids, oxidizing agents, ionizing radiation, all kinds of things that were classical identifiers of nucleic acid-based viruses, RNA or DNA viruses, they have no effect on this phenomenon. These mice this intracranial injection will still cause a CJD-like phenotype. But if you treat these same homogenates with things that destroy proteins and have no impact on nucleic acids, you can completely inhibit this transmission. Here's the even wackier part. If the mice that you inject the brain homogenate into, if you engineer these mice so that they are PRP homozygous null for the endogenous protein, they're immune. They never get the disease, right? So this and a whole family of experiments, a lot of which that Prusner had to battle hard to convince people of, ultimately proved that this type of disease has nothing to do with a classical virus. This clear transmission of replicated information does not depend on a nucleic acid. It depends on only one thing, and that is the conformation of the PRP protein, right? In either a normal state or in the state. In other words, the disease-forming agent is an altered conformation of this PRP protein that can propagate itself. Right? That not only scared the crap out of people, because how do you go detect something like that? It also sort of shook the tree of central dogma of biology at the time, in the late 70s and early 80s, which said that information only flows one way. It flows from nucleic acids into protein, and that you got to have nucleic acid in order to propagate replicated information, right? To even begin to rationalize an experiment like that, we've got to understand a lot more about protein structure, and that's where we're headed next.